Frances Ashcroft is a professor at the University of Oxford and was central to the discovery of how insulin is released from the pancreatic beta cells. Frances was also involved in the discovery of one of the main causes of neonatal diabetes, a form of diabetes that affects infants on their first 6 months of life. Thanks to her work, individuals with neonatal diabetes were able to switch from taking frequent insulin injections to taking anti-diabetic drugs orally once or twice a day. Among her many accolades, Frances was elected a Fellow of the Royal Society and was awarded the Croonian Medal and Lecture by the Royal Society, one of the most prestigious prizes in the biological sciences. She was awarded the L'Oréal UNESCO Award for Women in Science, and in 2015, Frances was appointed Dame Commander of the Order of the British Empire by Queen Elizabeth II for services to medical science and the public understanding of science. In addition to her scientific accomplishments, Frances is an acclaimed author. She has wrote a textbook on ion channels and has written 2 popular science books, Life at the Extremes and The Spark of Life. If you're interested in hearing more from Frances, be sure to check out her podcast, A Good Science Read, where she and her guests discuss their favorite popular science books. I am Dr. Pedro Diaz, and this is the Screw That Podcast, episode number 4. I'm here with Frances Ashcroft. Frances is a professor at the University of Oxford and has spent a long part of her career elucidating the mechanisms by which insulin is released from the beta cells in our pancreases. Not mine anymore, at least, but from those who don't have type 1 diabetes like me and have a functional beta cells in their pancreas. So, Frances, I was introduced to your work in 2018. This was when I was doing my postdoc at the University of Pennsylvania in Philadelphia, and I was working at the Department of Physiology, which has a long tradition on the study of ion channel biology. So Carol Deutsch is someone who I have very fond memories of, Clay Armstrong, Toshinori Oshii, Kevin Foskett, and many others. So it was by that time that I came across your review paper on the electrophysiology of the pancreatic beta cell with Patrick Rorsman. This was published in the Journal of Physiology Reviews, and I just found it to be such an entertaining and informative read. You know, living with diabetes myself, it was really just a fascinating exercise to travel through the history of the wonderful discoveries about how the beta cell works, and specifically the mechanisms that mediate insulin release. Discoveries in which you played a central role and that we're going to talk a lot about today. I would love to start by asking you to tell us a little bit about your background, your path, and what led you to perform research specifically on pancreatic beta cells.
Well, I grew up in a tiny village in the middle of nowhere in the county of Dorset in England. Went to the village school, which never had more than 20 kids in it, and the teacher was my mum. And I spent most of my time running around the fields, being a naturalist, looking at wildflowers and birds and things. And that's what I always thought I would be. I thought I would be some sort of naturalist in my life. But then I got fascinated by science, and I was fortunate enough to go to Cambridge University, where I read zoology, and I worked on stick insect muscle fibers for several years in my PhD, and nobody really understood why it was interesting to study stick insect muscles. So when I got my own lab here in Oxford, I decided that I would work on something different. I wanted to use a new technique that had just been invented that allowed you to look at the currents that flow through single ion channels when they're open, so you can dissect out their properties. And I deliberately sat down and decided which cell I was going to pick to work on. And I decided it had to be something which had a disease associated with it, so people would understand why it was interesting to look at. It might be easier to get funding for, always a problem. That there would be somebody in Oxford who was working on the same area, but not using the same technology as I was. And it seemed that diabetes was the perfect thing to work on, because there were people here working on the biochemistry of insulin secretion, and there were people who obviously were working on clinical aspects of diabetes. But nobody was looking at electrophysiology or ion channels. So that's how I decided it. I just sat down and very specifically decided this is what I wanted to work on.
And then what happened then? So this was like you joined Oxford back in '83?
Yes, about that time. Well, it was funny, of course, because I've never thought that I was any good at anything. And I was mad. It was a completely new technique known as patch clamping, which allows you to look at the activity of a single ion channel, which I'll explain in a minute what they are. And I'd never used it. There were only 3 people in the UK using it. It was basically learning as you go. And there were not, it had just been invented a few years previously. So I think I was somewhat too adventurous in a way. And then the other problem was it was thought that one should work on a cell line because they would be easier to operate, to do the technology. So basically, this is what happens. We know that if I explain what ion channels first, and then I'll say something about how we study them.
Go for it.
So
Go for it.
Every cell in your body has a membrane around it that acts as an interface between the inside world and the outside environment. But obviously, things have to get in and out of the cell. There are transport proteins that enable that to happen. And one of the type of these transport proteins are the ion channels, which are basically no more than little gated pores that sit in the membrane and they open and close. And when they open, ions can go through, things like sodium or potassium. And when they're shut, they can't. And it was very clear from the work that had been done previously that an ion channel had to be involved in the way in which glucose stimulates insulin secretion from pancreatic beta cells. So I set out to look for that. And having read the literature, it seemed that it was likely this was going to be a potassium channel. That meant it had to be closed by glucose somehow. So that's what I set out to look for. And the way that you do this is to take a single cell and to bring a glass pipette down onto the surface of the cell and form a high resistance seal between the glass wall of the pipette and the membrane of the cell, so that you isolate just 1 or 2 ion channels underneath. And then you can record the incredibly tiny currents that go through this channel when it's open. They're 4 picoamps or so, and it takes, 3 amps to run your electric kettle. So that's very, very, very much smaller. And so I set out to do that. And it was very funny because I managed to record some channels, but they were never affected by glucose. And I was getting the cells from, this was another funny story. I needed cells to look at. So where was I going to get these beta cells? And it just turned out that there was someone in Oxford who had a cell line of beta cells. So he had tissue cultured beta cells. And his name was Stephen Ashcroft. And I thought this would be really rather amusing to have a paper, Ashcroft and Ashcroft. What I hadn't anticipated is that this caused some confusion in many circles because people assumed we were married, which of course we weren't. Anyway, Steve and I decided to work together on this cell line with him providing the cells and me doing the electrophysiology. But I never saw any effect of glucose. And I was talking to Steve's postdoc one day, and she said, but the cells don't respond to glucose. You have to look at real beta cells, isolated from a mouse, to be able to see an effect of glucose. And she had never isolated any single beta cells. The whole islet, as you know, beta cells come in islets with maybe 1,000 cells or so in them. But she had never dissociated them into single cells. So she gave it a go. It was amazing because that very day, I found the first closing of this potassium channel by glucose on these native beta cells. So that's how it all got started.
Wonderful. And then what were the next steps? Like if we were to translate that to the classical example of...
so how did it work?
Yes.
So what happened was, what was already known was that glucose had to be taken up and broken down, metabolized, to stimulate insulin secretion. And it was also known at the time that a rise in intracellular calcium ions was responsible for triggering the movement of the secretory vesicles that contain insulin to the surface membrane, where they fuse and release the insulin into the bloodstream. But how it was that metabolism should somehow or other cause calcium entry through calcium ion channels. That wasn't understood. And that's what we were able to fill in, that little bit in the jigsaw, because what we were able to show was that the potassium channel sitting in the membrane was normally open, and that kept insulin secretion switched off. But when metabolism was increased by breakdown of glucose, it produced a chemical called ATP. That's the energy currency of the cell. But in this case, it was acting in a different way. It was binding to the ion channel and switching it off. And that meant that resulted in the opening of the voltage-gated calcium channels, calcium coming in and insulin being released. So we found this little bit with the metabolism regulating the ion channel.
Precisely. So translating that again to the classical example, when we eat, a human eats, this is essential. The nutrients start to get absorbed. There is a raise of glucose in the bloodstream, and then glucose goes in the beta cells, and it starts to be metabolized. And that was the piece of the puzzle, this really, really important piece of the puzzle that you and your lab found out. And so if we turn this classical example around, how is this process regulating, for instance, during times when we are fasting, like for instance, when we're sleeping?
Well, it's all relevant to the blood glucose that is in your blood. So if your blood glucose goes lower, then what will happen is there will not be so much metabolism in the beta cell, and the channels will open, and insulin secretion will be switched off, which is what you want. Because, the really important thing, of course, as you so very well know, is to keep your blood glucose constant. Because if it goes too low, just for a few minutes, then the brain is starved of fuel. And obviously this is not good for it.
Exactly.
But on the other hand, if it's too high for too long, then you can get the complications associated with diabetes, like retinopathy and so on.
Exactly. And I think it's wonderful, those examples, because another example that comes to my mind about the beta cell and how it just beautifully works is the fact that there are genes that are shut off on those cells. Like, for instance, two that come to my mind and they are relevant for exercise are like the lactate dehydrogenase, and there is another transporter, doesn't allow pyruvate and lactate.
This brings us to a sort of different story, which is the story of metabolism itself. So the beta cell has an unusual metabolism, which is because it is specialized for sensing extracellular blood glucose. So the two things which are important, first, glucose uptake. Glucose just comes into the cell, so the extracellular glucose tracks the intracellular glucose tracks the extracellular glucose, and then it's phosphorylated. And this is done within an enzyme called glucokinase, which is specialized in the beta cell because it can respond to a very wide range of glucose concentrations. So it's sensitive across the whole range which you might normally experience. That's important because it means that a beta cell is able to respond to changes in blood glucose with changes in insulin secretion.
And it also doesn't get inhibited by its own product.
That's also important, yes. So, and then the other thing, of course, as you've already mentioned, so if we think about metabolism, which is complicated to describe, the glucose is kind of metabolized in two stages. The first is known as glycolysis, and it takes glucose to pyruvate. And when it becomes pyruvate, then it enters the mitochondria, which are these powerhouses of the cell, where it is further metabolized to produce a lot more of ATP. So much more ATP is produced in the mitochondria than it is in the glycolysis. And it's very important in the beta cell because all the sugar goes into the mitochondria and is metabolized there. So it goes straight through to pyruvate and then into the mitochondria. What happens in many cells is that some of the pyruvate I should say, the mitochondria don't always manage to keep up with all the glucose that's coming through. And in that case, the glucose from the pyruvate can be channeled to lactate and then leave the cell. But this doesn't happen in the pancreatic beta cells because what happens is they have some lactate dehydrogenase, but not very much. But more importantly, they don't have the lactate exporter. And this is crucial because you don't want, when lactate rises during exercise, you don't want it being metabolized by the beta cell and producing insulin, which might lower your blood sugar just when you need it.
Precisely. So you just touched on a broad range of factors that are involved in the release of insulin from the beta cell to the bloodstream, such as glucokinase, the potassium channels. So in my case, as someone with type 1 diabetes, all of these factors were working properly, as far as I know. Then, through an autoimmune process, when I was probably around 20 years old, I started developing antibodies against my own beta cells, which led to their destruction and by my own immune system. However, there are other types of diabetes that result in a loss of control of blood glucose levels and hyperglycemia. So without the autoimmune destruction of the beta cells. So for instance, type 2 diabetes, which is the most prevalent form of diabetes in the world, and neonatal diabetes, which is relatively rare and occurs typically within the first 6 months of someone's life. So tell us, how did your work inform us about the development of these other forms of diabetes, such as type 2 diabetes or neonatal diabetes?
Well, I have to say that when I first saw the glucose close the KATP channel, I thought that this was what caused diabetes. I thought if glucose didn't work, if glucose was unable to close this channel, then all of the subsequent things wouldn't happen. There would be no insulin secretion and the patient would have diabetes. So I was incredibly excited that evening. But it didn't turn out to be quite like that. We spent some time looking for mutations in people who had type 2 diabetes to see if we could find something that was associated with the disease. And we looked at 100 patients and we found a common genetic variant that wasn't quite statistically significantly associated with type 2 diabetes, and we didn't find any unusual mutations. So that idea went out of the window. But one should never completely lose those ideas. And what happened was we then spent years and years, not just us, but lots and lots of people around the world studying this ion channel, how it was regulated, because it isn't just ATP that regulates it, there are other things as well. And then we tried to clone the channel, tried to get the DNA for it. And that was very difficult because it turns out that the channel is made up of 2 different types of protein, and you needed both of them to make the channel. And so there are 2 subunits, two main proteins, one which forms the pore and one known as the sulfonylurea receptor, which is a regulatory subunit that binds the anti-diabetic sulfonylurea drugs, which act by blocking the channel and stimulating insulin secretion. It's another story. So a group in the States, the Bryans, got the sulfonylurea receptor, and we were still looking for the other one. and finally, we and other labs got the second subunit, put them together, found a channel. So then, of course, the next thing was twofold, really. Which bit of, which subunit does what? Where do the drugs bind? Where does ATP bind? How does the channel actually work? That was one thing that we were looking at. And then the other thing was, of course, now that you have the genes, you can look and see whether in either of those genes there are mutations associated with diabetes. And as I said, we looked, but we looked in 100 people, and that wasn't enough. And then my postdoc and friend, Fiona Gribble, said, Fran, we're looking at the wrong people. We should be looking at people born with diabetes if we want to find severe mutations. And of course, she was right. So I went back to my clinical colleagues and I said, could we have some DNA from people born with diabetes? And they said, they don't exist. Well, that was wrong, but I believed them. It just is that they're very, very rare, as you said. And then my wonderful colleague, Andrew Hattersley, made a collection of DNA from people who were born with diabetes, and DNA from people from all over the world. And he started screening them for mutations in the pore-forming subunit of the KATP channel. And that was another red letter day for me, because I've never forgotten the day he phoned me up and he said, Fran, I think you should sit down. I've got something exciting to tell you. And then he told me they'd found the first mutation, and he asked if we would like to collaborate with him and look at its function. And of course, we found that the channel was much less sensitive to ATP, and it wasn't closed by ATP concentrations that would turn the channel off. So that was how those patients had diabetes. And we also found that the channel was sensitive to sulfonylurea drugs, which bypass the metabolic steps and act on the channel to close it directly. And of course, that immediately suggested another possible therapy for these patients, that they no longer needed to have insulin injections. They could have sulfonylurea pills.
I think that's such a wonderful example because, I mean, these patients, they were chronically dependent on insulin injections. And I know what it is to live like that. And just the idea of having to just take a pill, essentially, every day and things get better is just wonderful. And it's such a cool thing to think about because sulfonylureas, like, they were developed during the Second World War, right, to treat infections. And then there was from what I recall, a French doctor who noted that some class of these sulfonylureas were linked to hypoglycemic episodes in some patients. And then another French doctor, I think, he discovered that they were linked to an increase in insulin secretion from the beta cells.
Yes, in dogs.
in dogs, and then eventually this effect was very interesting, and then it led to the re-engineering of these molecules to kind of like take away the bacteriostatic effects of them while retaining the hypoglycemic and anti-diabetic effects. And I think the first one, from what I can recall, is tolbutamide. Or so how was that, like, back in 2004? This. Starting to treat patients like this?
Well, of course, I never did any of that. That was done by the patient's doctors.
Sure.
First, in places like the Netherlands and Norway and so on, people who were collaborating with Andrew and myself. I should say that the drugs are very effective on the insulin secretion, but it's important to remember that some of these patients have very severe mutations. And those people who have these types of mutations, they not only have diabetes, they also have neurological problems. And unfortunately, it seems that sulfonylureas don't get into the brain in sufficient quantity to be able to reverse those effects. Or maybe they get in there, but they're unable to reverse them because the KADB channel was required to be operative during brain development. It was clear, I think, for me, one of the most exciting things was that it seems that the drugs can help motor development, help the children walk. Some of them couldn't walk before. And I think that was, for me, one of the really exciting things, to be in Topsham, down in Devon, where Andrew is, and to see a video which was brought over by Brunning, from the Netherlands, of a little boy who had been unable to walk, but a month after taking sulfonylureas was running around and playing football, as well as his diabetes being cured. And that sounds great, but unfortunately, some of the other problems are not affected. It's great if you have a mutation which isn't very severe. And of course, the other thing is, people have been taking these drugs now for 20 years or so, but we don't really know how long they will last for. But so far, 20 years is 20 years.
In general, they are reacting well in terms of long-term treatment with these drugs?
I think it's more difficult around, teenage times. I think that becomes more difficult. They tend to have to take more drug. But as I said, I'm not a clinician. I'm just a scientist.
And as a scientist, how was that experience? I think few scientists go through, even with the long careers, they go through that moment of having such an impactful discovery on people's lives.
Well, we were just very lucky, and I think for me, one of the most wonderful things was because it was so rare. There were not many people, people didn't know anybody else who had the same problem. So Andrew and I decided to hold a conference for them in London. And that was really interesting for two reasons. First of all, because it enabled us to meet the patients, but it also enabled them to meet each other. And that was really important for them to, so what do you do when, for example, when your child has got gastroenteritis or something, and they're taking a pill? How do you control their diabetes, etc.? And just to learn a little bit more about some of the other issues that the patients with the severe mutations had, which was also possible at that meeting. And now they've formed Facebook groups and all this sort of thing. And there've been regular meetings in Exeter and in the States and so on.
Your scientific journey is a wonderful example of how the elucidation of a basic biological mechanism can really bring about radical change and improvement of care in humans. But it really goes beyond that. And it was one of the groundbreaking discoveries that helped tying metabolism with electrical activity. And you wrote a wonderful book about this called The Spark of Life.
Oh.
And so in this book, you really beautifully illustrate how electrical activity is everywhere in our body. And besides the beta cell and the secretion of insulin, what are a few of your favorite examples of electrical activity in the human body?
Well, I mean, they're all interesting. I happen to have a passion for ion channels, and ion channels are found in every cell of our body, and in every organism on Earth. It doesn't matter whether you're a bacteria or a giant sequoia, they are there and they're doing things. And I think that you should think that they're operative even before conception, because I always like to say that the great sperm race is one of the races that each and every one of us has won. And it is true. And we do it with the aid of an ion channel called catspur, which kicks in at the last moment and makes the sperm beat even faster so that it can actually penetrate the egg. They're everywhere. They're why your nerves and muscles work, obviously. They cause the electrical activity that's taking place in your brain right now. They're in every one of our sense organs. I think some of my favorite stories, though, are what happens when things go wrong. So, for example, there are so many extraordinary toxins that work on ion channels. I think everybody's heard of the pufferfish poison, tetrodotoxin, which, of course, if you think about it, we all know somebody who's been poisoned by tetrodotoxin. His name is Commander James Bond. And he emerges shaken but not stirred from his encounter with the blue-ringed octopus, or whatever it is that has tetrodotoxin in it. It blocks the sodium channels in your nerve fibers, so it basically prevents you breathing, and you die of asphyxiation. And then, of course, there's other ones. I think another one I think is very interesting is a toxin found in the nectar of rhododendrons, which is taken by, bees make honey from this nectar, and the toxin is in there. It's called grey-anotoxin, and it works by opening the sodium channels too much. So it does the opposite thing. It was first reported, I think, by Xenophon back in 400 BC or something, when he was campaigning near the Black Sea, because it's found in the nectar of rhododendrons that line the edges of the Black Sea. And he reports how his soldiers, they're throwing up and they're extremely unwell, and they have heart arrhythmias and so on. And I think he had this wonderful phrase, they lay around on the ground in a state of great dejection. And fortunately, most of them recovered. But 300 years later, when King Mithridates was fighting Pompey in the same region, he remembered this story, and they went out to pick the honey and strewed it in the path of the oncoming troops, who ate it and were rendered hors de combat and easily massacred. So this was a sort of early form of chemical warfare.
Exactly.
So, there are all these fascinating things. And, well, they won Nobel Prizes really rather a lot. And the last one they won it for, if I remember correctly, is for the ion channels that are involved in why chili peppers taste hot. So these are ion channels that are sensitive to heat, but they're also sensitive to things like capsaicin in chili pepper. And there are lots of mutations in ion channels that cause all sorts of weird and wonderful diseases in humans and in animals. Maybe not so wonderful if you have them yourself.
Exactly, yeah. No.
But interesting from a scientific point of view, because they can tell you something about how the channels work, and they can also help you design drugs that target them. So an awful lot of drugs work on ion channels, local anesthetics, for example.
There are people who can eat chili peppers without feeling any sense of hotness.
I don't know about people, but certainly birds aren't sensitive to capsaicin. So if you want your squirrels to stop eating your peanuts that you put out in the bird feeder, the idea is to coat them with a bit of chili pepper, and the birds can eat them, but the squirrels don't like it. So I'm told. I have to say I've never tried it. In my experience, squirrels are remarkably clever.
They will wash the peanuts or something.
Probably, yes.
And so, Frances, And what is going on in your lab? Would you like to share some of your latest works and findings?
Well, the thing is, we've now started to work on type 2 diabetes. And what I wonder is whether in type 2 diabetes, it's not the channel that goes wrong. It is the metabolic regulation of the channel that might be impaired. And actually, you don't need to alter channel activity very much in order to inhibit insulin secretion. It can be really quite a small increase in opening. And so that might be that if your metabolism doesn't work very well, this is what happens. And what we have found is that if your blood sugar goes up just a little bit, it causes all sorts of changes in the pancreatic beta cell that reduces insulin secretion. So elevated blood glucose seems to be bad for the beta cell and bad for you. We have a mouse model of this, and in mice, the blood sugar only has to go up by 3 millimolar chronically for these changes to start to happen. Basically, what happens is the mitochondrial metabolism gets turned off. It becomes really quite complicated. Metabolism is horribly complicated. I'm just learning to be a metabolic biochemist. Basically, if you switch off the mitochondria, where most of the ATP is produced, what happens is pyruvate builds up. But as I've already explained, it can't go to lactate as it would in the heart or your muscles. So everything piles up above it. And one of the metabolites in the pathway above it then starts all these gene changes that turn off glucose, stimulated insulin secretion. And the other problem is the cells fill up with glycogen because there's nowhere for the glucose to go. Normally, beta cells don't have glycogen because this isn't a very good idea, because obviously they don't need a store of glucose in the cell, because what you need them to do is to be able to respond to changes in extracellular glucose. There's nowhere for the beta cell to put it. So at least in the mice, they put it in glycogen. And I think also in human type 2 diabetes, similar things are going on, because using donor tissue, we have seen glycogen particles in human beta cells from a person who had type 2 diabetes.
That's incredible. So the implication is that even when a human has normal or low blood sugar levels,
not normal levels, but slightly elevated. But 8 millimolar glucose in your blood long term is not good for you.
Sure, but the beta cell will still respond to the glucose because the glucose is literally inside the cell, or the glycogen can be broken down to glucose, and the beta cell.
The trouble is, I think it doesn't work very efficiently because it's not making as much ATP.
I see.
So anyway, what the results suggest is that so far, and this is speculation now, right? So the speculation is that there's too much glucose coming into the beta cell, and that is messing up metabolism. If, for example, we could reduce the amount of glucose that comes into the cell by lowering your blood sugar concentration, or perhaps by reducing metabolism at a stage upstream of this metabolite, which is causing all the deleterious effects, then that might be helpful. So those are the things we're sort of looking at. But it also makes me think, glucokinase activators have been tried as drugs in the past to stimulate insulin secretion. They work in the short term, but they never worked in the long term, and they also caused liver problems. And I think one of the reasons this might be the case is because basically it's like just shoving more glucose into the cell, which isn't very good for it. Anyway, that's what we're working on.
Yes, I think, correct me if I'm wrong, but is it Elizabeth Haythorne also researching something on that?
Lizzie, yes, absolutely. She was a postdoc in the lab until recently, but she's now set up her own group in Edinburgh, which is wonderful. And so she will be carrying on the work and also doing new things, her own ideas.
Sure, wonderful. And on that note, you've had such a massive impact in the field of diabetes research. Your legacy is really simply remarkable, and a big part of this legacy are the people who you trained in your lab and went on to establish their independent research careers.
That is a great joy of doing science.
Absolutely. So from the top of your head, do you know the number of scientists you have trained during your career?
No.
and so
50, maybe more than that. I don't know. There have been PhD students and there have been postdocs. Yeah. Sometimes the postdocs have trained me.
I think that's also like a really great
And of
experience.
course, also the other thing is one shouldn't forget that one also has wonderful collaborators.
Sure.
So there was Patrick Rosman, we've collaborated for more than 30 years. We started out as competitors because I got a paper from Patrick to review immediately after I discovered the glucose sensitive channel. And I got the paper from Patrick that said, and some people in the States had found a channel that was sensitive to intracellular ATP. But it was at very, very low concentrations. So you wondered, it would always be shut in the beta cell. And then along comes Patrick and with a paper that says the glucose sensitive and ATP sensitive potassium channels are the same thing. Well, I had exactly the same data, but Patrick's paper was here sitting on my desk and I had to review it. So, of course, I accepted it and said it was a wonderful paper, which meant that my results went in the bin. So you can imagine when I met Patrick in a swimming pool in Alicante at a conference, I was not at all pleased by this young man. But we decided it was more fun to collaborate than compete. And so we've been collaborating for 35, 40 years. I don't remember how long. He's brilliant. And then, of course, Steve Ashcroft I collaborated with at the very beginning. And then there was Andrew Hattersley and many more. I think it's one of the great joys of science. It's wonderful, all the people whom you meet.
How do you perceive your own legacy today? Like, for instance, when you see a former PhD student or postdoc of yours publishing their papers?
Well, I think it's brilliant. I'm excited at what they do, but that's their work. It's got nothing to do with me. I'm just happy that they're doing well.
You just mentioned like a situation where, it's really difficult to deal with, right? You have this amazing discovery and then there is someone else who did the discovery and supposedly is one step ahead in the sense that you have the paper there. Like they are already,
yes.
With the paper submitted, the work is finalized essentially.
Yeah.
So this is also like a common theme on the conversations that I have with the people who I have invited so far here, and it's the resilience and the overcoming of the challenges that life throws at us. So would you like to share some of the challenges like that, just like that, and how you actually, the ways you faced them and dealt with them? In this case, it seemed like a wonderful way because you actually started to collaborate with Patrick Rosman, but just
it doesn't always happen like that.
Yeah.
I can't say anything better than keep calm and carry on. The famous poster. I learned over the years that the data that you have can also be used in a later paper that shows more. You just have to do more work. I think what's always saved me is being interested in the science. I've just wanted to know. And if you're fascinated by the science, it kind of grabs you and you can't stop. So you carry on. I think it's always best to be generous and collaborative. In my experience, that works much better than being, difficult. I think in my case, of course, the most problems I had were later on being female. It wasn't so easy getting a job sometimes. But that's all changed now. It's not a problem.
Frances, you've published two popular science books, The Spark of Life and The Life at the Extremes. Are you working on a new book? I would love you to be.
Not yet. No, I'm trying to think what I might write it on. But you see, the problem is when you look at the book that you've written, when you write the book, you think, I can't possibly write this. And I looked back at the first one, Life at the Extremes, and I thought, how on earth did I write this? It's actually it's not that bad. After 10 years, that's what I thought. But I can't possibly write another one because I'm half senile now, so there's no way I can write the words. And then, of course, I wrote the second one. Now I look back and I think, no, I'm really failing. So we'll see whether I get one out or not. I mean, it is one of those things. It's difficult. It's very hard work, and it's difficult to get started. And you always feel, probably AI can do it better now.
I doubt AI could do better than you. And I would say I would love to have a third book from you, but if you somehow are not able to start for some reason, maybe an update on The Life at the Extremes, because some of the examples there, like the marathon world record has been beaten.
So you broken.
Yeah. So the humans keep pushing the extreme, and so perhaps just an idea, an update.
Yes, but I think it'd be more fun to write something completely different. I have a few ideas, but we'll see.
Looking forward. And you have a podcast
I do,
where,
yes.
yeah, and where you invite
I'm not sure
guests.
that's any good either. I mean,
I've listened to it and I love it. So
Oh, you do?
yes. So you invite guests where, and then you discuss your favorite popular science books. It's called A Good Science Read. And Frances, what is the book that is currently sitting on your nightstand or on your desk?
Oh, well, I suppose the last one is one that I've done. Is amazingly, we discussed metabolism. There is a book called Burn by Herman Pontzner, which is all about the Hasda and has this very interesting take-home message, which is that it doesn't matter whether you are a hunter-gatherer, tribesman who walks, 20,000 steps a day, never sits in a chair but squats down, or you're an American couch potato, well, maybe a UK couch potato, like me, I should say, and you really don't do anything. You burn the same number of calories a day. You have the same energy expenditure. It's just that what the body does is it moves around where it's spending the calories. So instead of spending it on exercise, it's spending it on immune function and things like this, because exercise is very good at reducing inflammation. And since many of the diseases of old age are inflammatory, it's very important that as we get older, we exercise. But it doesn't have to be running marathons. So there's that book, which is all about how we're using energy and is a very straightforward read. And then there's another one, which, would you believe it, is all about the Krebs cycle. And given that the Krebs cycle is quite difficult to understand, it's a major triumph to have written a book all about it. But basically, he says that this metabolic pathway, which we use to burn fuels in oxygen to make energy, actually worked initially the other way round. And when there was no oxygen, it was very important for making amino acids and sugars and things like this way back millions of years ago. So this is also a very interesting book.
That is interesting. So what's the name
So
of
that the name of the book is Transformer, and it's written by Nick Lane. And that will be the next podcast coming out, because we've already done it, but I'm just sort of tinkering with it at the moment.
Are you having Nick Lane there with you?
No. The idea is, unfortunately, well, I thought there were quite a lot of podcasts where people discuss their own books, but this one is slightly different. This one is where I and my guest discuss two books, one chosen by them and one chosen by me. But you can't choose your own book, and you can't choose a book the other person's written.
I was asking because I was fortunate enough to be at UCL, and I've listened to Nick Lane talk about the origin of life, and he's just like such a wonderful person to listen to, so knowledgeable. So.
Well, perhaps I'll have to change it and start interviewing the writers as well. But this book is a real work of scholarship. It's a really important book, not just entertaining. Frances, so I recommend them both.
Yes, I will put them on my list. Frances, thank you for your time. It was really a delight to speak with you, and thank you for all you've done to advance diabetes care and improve the lives of people really all across the world. Thank you so much.
It's been my pleasure, and I'm sorry that we've not been able to do anything about type 1 diabetes.
It will get there eventually, some other people.
Exactly. Thank you.
Thank you, Frances.